gotten the seat, don't forget to bring that with you to lab today. This is a way to do like little miniature streak plates without, you guys are trying to get between 20 and 25 different looking bacteria out of your soil sample. So there's 11 lab groups. So if you each use like 5 plates, then that's like 60 plates. If you each use 25 plates, then that's like 1,000 plates. And so we do the quad streak. This bacterium is called Bacillus mycoides. And as you can see, it does not stay where you put it. So they put it here, and it does this like totally awesome, but a bit unwieldy, tendril growth all over the whole plate. If you have this one, do not put it in the quad streak plate. It must go on its own. If you see any other ones that kind of look like they're moving, then also put them on their own. Because if you put them on the quad streak, then they'll grow over the whole thing, and then you will have lost the 3 other ones that were separated on there.
What was it called again?
Bacillus mycoides. I mean, you don't need to know what it's called, but it's really common in the soil. Ooh, this reminds me, I had a few people ask me in the reading for fungi, there were a bunch of different organism names. You need to know none of those. So what you need to know is on the things to know sheet. There will be, later on in this semester, some specific organisms that you'll need to know, but then those will be on the things to know sheet. Because microbiology is like all of the biology that you've ever learned in one cell. So there's no doing all of it in one semester. So we're just like trying to hit things that seem important or things that you're likely to come back to in other classes or in future school or whatever. And so the names of the fungi, well, nifty, you do not need to know. So get out your clickers, and we have a couple of questions. You guys all ready? Everybody on Finley? The question. That's 6. Wait, it doesn't even have to. That's weird. Let's try that again, shall we? Now it should do that. There we go. Just like it wasn't. Okay. So the distinguishing part of this question is short term. So carbohydrates are more long-term energy because you have to break them down. And then what is the short-term with short-term energy currency in the cell? ATP, which is a nucleic acid. Which nucleic acid is it? RNA. Excellent. So if it's the triphosphate, yeah, if it's the triphosphate, it's free, like when it's built into the nucleotide. So yes, and I mean it would match with TT, like the T, the A and the T would match, but because the TP is on its own. Okay, how did you guys do on this? Oh dear. Oh dear. Okay, ribozymes are catalytic RNAs. They are not enzymes. No one writes enzymatic RNAs. That is not a thing. They're catalytic RNAs. Where in the cell now, today, do we find ribozyme? / which is the most famous ribosome? Ribosomes. So ribosomes, this is a ribosome. The blue parts are the RNA, the ribosomal RNA, and the green parts are the protein. So this is the blue parts on their own. It is the ribosomal RNA that actually binds the mRNA, binds the tRNA, and makes the amino acid, the reaction that joins the amino acids, form. So the making proteins in your cells is actually done by a catalytic RNA, which is called what? Ribozyme. In the ribosome. So people were super excited when they found these, because what do we already know about RNA from the ATP clicker question? Right? It's a nucleic acid, but what can it do? It can do short-term energy. Okay, so what else do we know about RNA from like regular things that happen in the cell? What else can RNA do? It can go outside the nucleus, carrying with it information to do stuff, specifically make proteins. And so we've got the energy and we've got the information, but you guys know, like, we can't really make cells go without enzymes, like something to make the activation energy a bit lower and make the reactions happen. And so when ribozymes were discovered, scientists were super excited, because once we have ribozymes and we can catalyze reactions, then this provided enough evidence that you could get the basics of life with just RNA. Because you can have RNA to carry the information, RNA to provide the energy for the reactions, and RNA to catalyze the reactions. Different forms, but if all of this happens at the same time together, you can have life just with RNA. Does that make sense? This is called the RNA world. Do you guys remember reading about this in the book? So not only are ribozymes able to help in the formation of proteins, they also can copy short pieces of RNA. And so that suggests that they can be self-perpetuating and then possibly have led to the first cell. So now we have DNA, which helps with the information, and we have proteins, enzymes, which help with catalysis. But RNA still does all of these things in our cells. And so there's some idea that at the very beginning, before there was DNA and before there was protein, there was a single molecule, a single entity that did all of this, and it was RNA. So this is a really, really popular and common theory. So I wanted to make sure that you guys had heard of it. Any questions about that? Okay, great. Here is a clicker question for you. You guys can chat during the clicker questions. I mean, it's not like a test, you know, like, if you want to chat, you can chat. You still only get 30 seconds, but you can chat. Excellent work. So panspermia is meteorites. What's the other created from scratch on their own? De novo. Fantastic. So the other awesome thing about RNA is people have been able to create it from scratch in the lab. The slight problem is they can only make G's and C's. They can't make A's and T's. But they can make G's and C's, and that's better than being able to make nothing. So it could have been a combination, or like panspermia is probably the more well-supported idea, because the Big Bang did create huge numbers of diverse biomolecules that were on everything. And so then if the meteorite splashed in the ocean, then everything's floating around and it can get together on the charged clay surfaces and then make the membranes like in that video. And then if you get a little piece of RNA inside of a membrane with some hydrogens on one side, then you have something that looks kind of like a cell. So that's the idea. Any questions about that? Okay. So I'm going to take I didn't realize that this was a couple of slides to tell you about phylogeny. Phylogeny is the historical relatedness, and it gives you those like branching tree diagrams. So all phylogenies, nope, that's a lie. All phylogenies that include multiple diverse life forms, so the tree of life, we're showing how archaea, bacteria, and eukaryotes are related, or showing how mitochondria are related to bacteria, or anything that compares diverse life forms, uses the ribosomal RNA, the ribozyme. So in prokaryotes, because the ribosomes are different sizes, the ribosomal RNA is different sizes. So in prokaryotes, it's the 16S size, and in eukaryotes, it's the 18S size. So those sequences are compared. Really? Just going to do this. Take that slideshow. So you take your 16S and your 18S ribosomal RNA, and now that we have DNA, we are not actually looking at the RNA. We're looking at the DNA that encodes the RNA that makes the ribosome, that makes the protein, right? So we say we look at the 16S rRNA, but really we look at the 16S rRNA DNA. But that's just a lot of confusing letters. So that's why we don't say that, but we really look at the DNA. So we align the DNA sequences, look for numbers of differences, and then build a tree like this, or sometimes the lines will come out like more of a V shape. Each of these branch points here are called nodes. And each of the nodes represents some sort of significant change in the sequence, in this case. Some sort of significant change in the organism or the sequence. The other super important thing about nodes is that you can flip the tree at the node. So C is not actually closer to D, even though right now they're together, because you can flip this. And also, if you trace like the total path length, C is equally related to D and E. So just because they look like they're close together does not mean that these two are more related. Does that make sense? So using those 16S and 18S rRNA genes has allowed us to make what's called the tree of life. This is a super, super simple version, and Luca is right here at the base of the tree. This is a slightly more complete version, and I love this version because it shows you how much of the organisms are bacteria, how much are archaea, and how much are eukaryotes. That like arrow is us. So it's just really cool, like how much is bacteria and how much they're doing. Yes?
What part of taxonomy is recorded on the ring? Is it like class, order,
These are individual species.
Okay.
Yeah. not all of them have had their rRNA gene sequenced, so these are all of the ones that had been sequenced when the tree was made, put together in one giant tree. So trees like this that show all three groups are called trees of life because they represent all of the diversity. Any questions about trees? No. All right. Excellent. So we're going to talk briefly about archaea and bacteria, and then, well, about eukaryotes. Alrighty. So this is a totally fantastic electron microscope cross-section of archaeal cells. What can you guys tell me about the ribosomes in archaea? They're 70S, made of Excellent. Okay, what can you guys tell me about the cell envelope? What is the cell envelope? What parts of the cell can be included in the cell envelope? The membrane, basically the membrane and anything else outside the membrane. So that can include the membrane, the wall, or archaea have something called the S layer. What can you guys tell me about the S layer? What's it made of? Protein. So this is important because in all of the other instances of things we call walls, the wall is made of what? Carbohydrates. So walls are usually carbs. So the S layer is made of protein. And it's a lot more flexible, which is why archaeal cells are weird shapes. Okay, the most substantial difference that sets archaea themselves apart. So they have some characteristics like eukaryotes. They have some characteristics like bacteria. The thing that makes them their own group, well, is their DNA, there's their rRNA sequence, but without looking at that, just looking at the cell, the thing that makes them their own group is their membrane. And so the archaeal cell, their membrane can be one layer or two layers. You guys remember that from last time? They can have the monolayer membrane. Then the structure of their membrane is quite a bit different. So this is archaea here, there we go. And their membrane subunits, rather than being fatty acids, are isoprenes. And they use an L-glycerol, which is the other way than a D-glycerol. And the isoprenes are connected to the glycerol by ether links. Versus bacteria and eukaryotes have the membrane that you're used to, which has the fatty acid, the ester link, and the D-glycerol. So if you look in like your intro textbook, it would have this structure. So archaea have a completely different membrane structure that sets them apart. So they have the isoprene units, and then they have the ester link. Oops, did I get that backwards? They have the ether link to the L-glycerol. And that's important because it sets them apart. And then what can you guys tell me about their DNA? Is it linear or circular? Circular, excellent. How is it compacted into the cell?
Usually histone.
Using histone. Where else do we find histones?
Eukaryotes.
In eukaryotes. So the circular DNA is more like bacteria, but the histones is more like eukaryotes, but the 70S ribosomes is more like bacteria. So the only thing, again, that makes archaea really distinctive on their own is their membrane. You can find archaea all over everywhere. Well, there's a couple of nifty things about them. Most methane-producing bacteria, so if you guys have ever heard, like, cows, the great air pollutants, right, because they, like, constantly are excreting methane. Most methane-producing organisms are archaea. They also can help with different nutrient cycling processes because they're able to live in really extreme environments. But I think, kind of the most interesting thing about them is that there are no pathogenic archaea discovered to date. There are some that live in and on us, but they don't cause disease. Any questions about archaea? Okay. We're gonna go. We're gonna go through bacteria. Kind of quickly as well. I guess I'll write them over here because we're going to spend the whole rest of the semester on them. So what can you guys tell me about their ribosomes? 70S, great. What is the ribosomal RNA gene that we use for sequencing in bacteria? What size is that? 16S. So they've got the 30S and the 50S. And then the 16S is part of the 30. So you guys will need to remember like all 4 of these numbers in conjunction with bacteria and archaea.
What was the 16S again? What is it?
So when we are doing sequencing to compare how things are related, that historical relatedness or phylogeny, we use the 16S, which is a subunit of the small subunit. So. Okay, what can you guys tell me about their cell envelope? The membrane is made of, has fatty acid subunits, which have a what kind of link? An ester link to the D-glycerol. And then, what kind of wall do they have? Peptidoglycan, excellent. This is the distinctive thing about bacteria. We only find peptidoglycan in bacteria. What can you guys tell me about their DNA? What shape is their chromosome?
Usually circular.
Usually circular. And how is it compacted in the cell?
Gyrates.
Right, DNA gyrates. This is also only in bacteria, but the thing that is most commonly used to recognize bacteria is the presence of peptidoglycan. Any questions about bacteria or archaea? Okay. If we are using the 16S ribosomal RNA gene to identify bacteria, which one do we use for eukaryotes? 18S. So you guys all have that like a couple slides ago. Okay, I have a couple of clicker questions. Excellent work. Question number 2. Fabulous job. Okay, now we're going to talk about how we got eukaryotes. So in general, in the eukaryotic cell, there's 2 different types of organelles. Organelles that look like other tiny things or organelles that do not. So the organelles that have characteristics of bacteria are the energy producing ones. So the mitochondria and the chloroplast and something called the hydrogenosome, which we are not going to mention. So the mitochondria and the chloroplast have several characteristics which make them look like tiny bacteria living inside of a big cell. All of the other organelles, which are connected as part of the intramembrane system, So the nucleus, the endoplasmic reticulum, the Golgi, right, like all of those membranes are connected, are all not related to bacteria. So those other organelles, the nucleus, the endoplasmic reticulum, and the Golgi, are thought to have just arisen by infoldings of the membrane. That is called the autogenic, self-formed. The nucleus, the endoplasmic reticulum, and the Golgi. So it doesn't actually show in this picture, but the endoplasmic reticulum is continuous with the nuclear membrane, and then you'll have vesicles that will come off of the endoplasmic reticulum and go to the Golgi. So those all work as a cohesive system, which is called the intramembrane system in the cell. And so those are all connected, and they all have different synthesis and processing jobs, but none of them are related to energy generation. I've just had a very odd realization. That's totally an animal cell with chloroplasts. How odd. Okay, moving on. So the mitochondria and the chloroplast look very much like bacteria and are thought to have come around when an archaeal cell, having some characteristics in common with eukaryotes, engulfed a bacterial cell and let it stay because the bacterial cell was making energy. So the endosymbiote hypothesis is when an energy-producing bacterium was engulfed by an archaea and gave us mitochondria or chloroplasts. So the specific evidence for that is, firstly, both mitochondria and chloroplasts have their own tiny circular chromosomes that encode things that are needed for the organelle but are not needed for the rest of the cell. Both mitochondria and chloroplasts have an RNA polymerase, which is found in bacteria but not found in eukaryotes. And then both mitochondria and chloroplasts have the 70S ribosomes inside of the organelle. So this is a phylogenetic tree showing you several different species of bacteria. And then this branch right here is mitochondria from plants, animals, fungi, and protists. So this shows you that using, what's the little piece of ribosomal RNA that we use for bacteria to make trees? 16S, excellent. So using the 16S rRNA gene sequences, we've been able to make this tree, and it shows us that the mitochondria match very closely to the sequences of the bacterial ribosomal RNA. Additional superficial evidence is the appearance. So this is a cyanobacterium and this is a chloroplast, and they both have this really distinctive membrane layering or infolding that you don't see in a lot of other cells. Yes.
So, according to this hypothesis, what happened to the wall of the bacteria cell?
Didn't need it. So the main job of the wall is to protect the bacterium from external stresses, like osmotic stress, things eating it, etc. So if it's now in another cell that's managing those things for it, it doesn't need the wall. So probably just lost it.
So if that's true, then mitochondria and chloroplasts are evolving on their own, just separate from cell evolvement, right?
Yeah, yes.
Since they're their own
Yes.
being adapted.
Yes. The other awesome thing about mitochondria and chloroplasts is they can come up, like divide and then fuse back together on their own. They will do it concurrent with the cell cycle, but they also will do it during interphase. So they can divide and then fuse back together on their own. As the cell needs more energy, so if you like suddenly start working out and your cells need a lot of ATP, your mitochondria will divide on their own, separate from the rest of your cell cycle. I know, isn't it? So any questions? Do you guys feel like you could explain to one of the freshmen in Gen Bio why we think that mitochondria and chloroplasts are related to bacteria? Yes. Okay, so you guys are good, you don't have any questions about this? Okay. Okay, now on to fungi. So I have a question for you, and then we have like the world's awesomest video. Okay, it's not the world's awesomest video. It's close. So I have 2 awesome fungus videos, and we can watch the other one next time, because otherwise, like, what do you have to look forward to? Oh, where's our right person? Not here. Excellent job. Do you guys know the word that means breaking down dead things starts with an S? Genius. Okay, here is this fantastic strawberry video. The other one is about ants. We'll watch it next time. So they have taken these strawberries and exposed them to Rhizopus stolonifer, which is black bread mold. And so fungi make their living by breaking down things, which means that they are awesome at destroying plant cell walls, right? You go in the forest, you see the rotting log. So the strawberries just become this mushy pile of strawberry juice. It's so excellent. Then, when the fungus has exhausted the strawberries and it needs to move on to a new food source, it makes the spores, which is what makes black bread mold black, is the black spores. And then those spores will either be ejected on their own or will be carried on like the wind or in your house, like the air conditioning air current or something, to hopefully for the fungus a neighboring food that they will be able to break down.
That is terrifying.
Yeah. But think how much gross dead stuff would be laying around without fungi. So that's a plus.
Do you have any idea how long it was? Oh.
No, people ask me that all the time, right? So then I should put it on the slide. Yeah, and this was obviously, I mean, they let it go for the whole time. They didn't get disgusted and throw it out partway.
So did they like introduce this fungi to the strawberries, or they just like set the strawberries out on a lot?
No, they specifically introduced this fungus. That's how they knew it was this. Right, but there are, as you guys know, right, you walk in the forest, there's like twigs and stuff everywhere. You do not see like lots of dead squirrels. Dead animals kind of vanish straight away. Dead wood hangs around. Plant cell walls are really hard to break down, and only fungi and some bacteria can do that. And so fungi as decomposers is a super, super important job for them. So I'm just going to tell you a little bit about kind of the basics of fungal biology, just looking at the slides. And then we'll get into what all fungi do for us next time. Okay, one of the super weird things about fungi, which people did not realize for a while, is that they will grow differently outside in the environment versus in your body. This is because you're hot, right? This is called thermally dimorphic. So morph is the shape. So at different temperatures, they have 2 different shapes, is what that's telling us. So this fungus is called Histoplasma capsulatum. It is really common in this area. And you can get it anytime that you're breathing in a lot of dirt. So if you're like tilling up your garden and not wearing a mask, or shoveling a dirt pile, or in a really dry and dusty cave or something like that where you're breathing in a lot of soil, you could be getting histoplasma. In the environment, it exists as, what do we call multicellular fungus? People that did fungus, starts with an M. Yep, super close. Those strawberries were moldy. Multicellular fungi are called molds. Does anybody remember this? If you were one of the people that looked up fungi? No one remembers it, okay. Multicellular fungi are called molds. So outside in the environment, histoplasma grows as a mold. It has these long fibers called hyphae. This is how it grows. In your lungs, histoplasma grows as a single-celled organism that buds, which is called starts with a Y. Yeast. Excellent. So yeast are characterized by their budding division. So multicellular fungi are called molds. Unicellular fungi are called yeasts. This is the same organism. If you take this one from outside and put it at a hot temperature, it will suddenly grow like this. So a mold versus a yeast is not necessarily a different organism. It's just a different way of growing. Fungi are really adaptable. They want to grow however they can grow to get the most nutrients. So that actually made identifying them really confusing, right? Because you have a doctor and he's like, my patient is clearly infected by a yeast called histoplasma. And then you have somebody that's like outside studying garden soil fungi, and they say, oh, well, I have this mold, I'm gonna call it Agelomyces. So for years, they had two completely different names. Nobody knew that they were the same organism. But histoplasma was the older name, and so when people figured out that they were one thing, it was the name that stuck. Okay, so the individual thread-like filaments are called, each one is called a hypha, and together they're called hyphae. The plural is hyphae. The mat of them together on the strawberries, that fuzzy mass, is called a mycelium. And that MYC part of the word is really distinctive for fungi. So anytime that you see that, the person who named that thing thought that it had something to do with fungi. So hyphae can have different internal structures. They can be what's called septate, where they have internal cell walls that divide, the hypha into individual cells, each with one nucleus. Or they can be cenocytic, where they have very few to no internal walls, and all of the cell contents, including nuclei, just kind of fish about places. And so you can have a bunch of nuclei together. These are, this makes it confusing to know if these are, yes?
I was just asking if that bacteria is called that because it does that branched filamentous behavior.
So I don't know, you guys are familiar with like diploid and haploid, right? So these are, well, either diploid or haploid, because that has to do with the number of chromosomes in the nucleus. So multiple nuclei per cell is called cenocytic. There's not, it's not like, I mean, they can still be diploid and be cenocytic. So just to make sure we're clear on that. When you look at the fungal cell envelope, the distinctive things about them are their membrane stiffener, which is ergosterol. This ERGO is also a term that is commonly associated with fungi, not exclusively, but commonly. So ergosterol, again, is a sterol, the forwarding structure that helps to stabilize the membrane. And then you can see that they've got a bunch of different carbohydrate portions to their wall, but the diagnostic one is chitin. You will also find, do you guys know where else you find chitin? That's keratin. Excellent. So in insects, shrimp, crab, lobster, all of those jointed appendage guys have chitin. But the only place that you'll find chitin and ergosterol together is in fungi. Any questions about this? Okay. I have 2 clicker questions for you. Oh, crisis sentence not normally right.
What is that ergo frequency on the rostral? What is the ergo?
I don't know what it means. I just know that it's really commonly associated with fungal terms. Excellent. Okay, so the whole mass is a mycelium. What's the hyphae? Each individual, so that's a hypha, that's a hypha. Each individual filament or thread-like structure is a hypha. The mass as a whole is a mycelium. Excellent. So fungi can reproduce asexually or sexually. In general, what are the pros of reproducing asexually? You don't need to find a mate. What else? Is it fast or slow? Fast. What are the disadvantages of reproducing asexually? Right, you're exactly the same as everyone else around you. Okay, what are the advantages of reproducing sexually? Diversity, excellent. What's the disadvantage of reproducing sexually? Takes energy, it's a lot slower, and you have to find a partner. Fungi will do both of these depending on availability of food. So if they can easily find food to eat around them, which one will they pick? I'm sorry, I didn't hear you.
Sexual.
No. Easily find food. Asexual. So if they can easily find food around them, there's no need for them to diversify. They can just move from food to food to food, quickly sprouting out new fungals, they're called spores, new fungal spores. And then they can eat the food and then make new spores and then eat the next food, and then as long as there's plenty of food around them. If there is no more food around them and they need to go to a new and unknown environment to find food, they will do what? Sexual. They don't know what will be in the new environment, so they want to maximize diversity, hoping that one of the spores will make it. So asexual reproduction is done by yeasts all the time, and that's the characteristic budding reproduction. The super confusing thing is that mold, asexual reproduction, is by spores. Where's my mouse go up to? So this is the gypsy moth caterpillar. It's the caterpillar that I worked on. And I worked on killing them using a fungus. because they were eating all oak trees and killing oak trees. And so you can see as the mycelium starts to grow through the body comes kind of plumped up, and then it shoots the spores off, hoping that they'll land on a new caterpillar. But there's tons and tons and tons of caterpillars in the forest, and so this is asexual reproduction. Basically, you take your mycelium with the nuclei, and it can be either septate or sinusitic, and you just chop up each nucleus like that, and those become your spores. That's how fungi do asexual reproduction if they make spores. The super confusing thing is that when fungi reproduce sexually, they also make spores. So unless you know that fungus specifically, you can't actually say what type of reproduction it's doing just by knowing that it makes spores. So the name of the spores for sexual reproduction varies depending on what kind of fungus it is. So yeah, that's mostly just informational. You don't actually need to know that. The important thing is that they can reproduce either way, advantages and disadvantages of each, and what would make them decide one or the other. And then just to let you know that spores are used in both. Okay, do you guys have any questions? All right, we're just gonna stop there for today, because then after that we get into business. So I'll see you after lunch. Please don't forget your quad plate directions. So what you guys are doing in lab today, right, is you're gonna look at your soil plates, you're gonna find all the ones that look different, you're gonna give them all a number, and then you're gonna streak them. So it's a very slow and tedious process, which I always feel is better to pre-warn you so that you don't get super frustrated.